Hi, science friends. I hope you all had a wonderful Christmas break and had a chance to recharge your batteries and are ready to jump back in with me today. So we are starting our unit in genetics. Genetics is one of my favorite units. Hopefully you had a little bit of exposure to it in middle school, but that's been a long time ago, so we will review everything. But let's see if we can tap into anything that you may already know. Also, if you don't have this in your packet, I dropped those off a week or two before we were out for Christmas break, so make sure to get those. Get these materials as soon as you can. If you're unable to get them, please let me know and I will help make sure that you can have access to these, the things that you need. Okay, so let's look at this. Using your previous knowledge of genetics, determine what maternal genotype would most likely yield offspring with such characteristics. So you see here we have our dad, and then we have our offspring. Now, you may not be familiar with this term genotype, but what a genotype is, it's the letters that make up your traits. Okay, so the dad's genotype is big B, little b, and the offspring is little b, little b. We always have to say big or little. Because otherwise, if you just say BB, I have no idea what you're talking about. So we know that offspring gets one of his letters, so to say. Those letters are called alleles, but he gets one of them from his mom and one of them from his dad. So let's say we got this first B here from dad, okay? So you see dad could have given big B or little b, but he gave little b, as shown here. So apparently mom also gave a little b. So what could the mom's genotype have been? You should say that mom could have been big B, little b, or mom could have been little b, little b. Which of those has a greater probability of giving the offspring like this? This one. Little b is more likely to have that offspring, okay? And so the second part of this question says to write a Punnett square, draw a Punnett square, using the genotype that we came up with for mom. So this is mom, I'm gonna put her on top, okay? And then I'll put dad down the side. Now you may not remember how to do a Punnett square, but they're really easy and that's okay. And we'll do a lot more practice of these. This is just to kind of tap in and see what we can unlock from previous years. So when we complete a Punnett square, basically we just move things from the top and from the side down to fill in the box. So this one's going to be big B, little b. This next one, pulling the b's over and down, is also going to be big B, little b. This one is going to be little b, little b. And this last one is also going to be little b, little b. So you see here we had a 50% chance of getting a big B, little b, as shown up here. And then a 50% chance of having little b, if that was the parents, okay? Now, one thing that's important to note, you could have switched mom and dad and put mom on the side and dad on the top and it would have worked out just the same, okay? But that is a Punnett square, and that's what we can do to see our offspring. So what we got, little b, there was a 50% chance that that would be the outcome, okay? The offspring also could have been big B, little b. Now, we don't know what those B's designate. This picture doesn't really tell us, or there's no information. Just looking at these, I think the father and the son look a lot alike, except for their eyes. So I'm going to guess it has something to do with the eyes, okay? The dad has a big B here, so that means he's going to show the dominant trait. And so here I would guess that the dominant trait is black eyes. The son has two little b's, and so that means he's going to show the recessive trait. And you may not know what these terms mean, and that's okay. We're going to go over all of that as we get to it. And so here I would say that the recessive trait are those red eyes, okay? And again, we don't know that for sure. I'm just guessing that from the picture, all right? Let's go ahead and jump into our lesson today. As we get started, I want to talk about a few important terms here to move us into our lesson. So the first is heredity. Heredity is the transmission of traits between generations. So you get the terms hereditary or inherited, all stem from that word heredity, how traits are passed down between generations. And so sometimes you may have heard before, I know when my little boy was younger, a lot of people would say he looked like his grandpa, that he had inherited those traits, from 2 generations above. And that's because those certain traits are coded for in our DNA, and that DNA is passed down between generations from parent to offspring, okay? And so variation is differences in traits between members of the same species. And so we see variation here in this eye color. You've got the red variant and the black variant. What are some other traits that you have that you see variation in? Well, you see variation in height. You see variation in freckles or no freckles. Maybe you can roll your tongue or not roll your tongue, or have a widow's peak or a hitchhiker's thumb. If we were in class, we would look at all of these things. Maybe you have attached earlobes where they kind of stick to the side of your face. Or maybe you have unattached earlobes where they're kind of dangly and they're loose there. Maybe even the ability to taste certain chemicals is an inherited trait. If we were here in class, and maybe when we get back we could do this one day, I would give you a piece of paper called PTC paper, and you would taste it, and the majority of you wouldn't taste anything, but the ones that did taste something, it would be very obvious that you tasted something there. And so those are variations, differences between members of the same species. And so genetics studies this. It studies these traits and how these traits are passed down. And it's important for a lot of families as they have children or are looking at, what's the likelihood of certain traits being inherited, and different things like that, okay? So as we study genetics, one thing that we're going to talk about later this week is meiosis. So previously we talked about mitosis, okay? And mitosis was the cell division that we talked about that happens in somatic cells, which are body cells. Meiosis happens in sex cells. And in mitosis, you make the offspring. You start with one cell called the parent cell, and when it divides, let me just draw a little diagram here. Mitosis. So you have the parent cell, and it has the full set of DNA, and when it divides, it forms 2 daughter cells that have the full set of DNA. So these are identical, okay? And it's 1 round of cell division, okay? Now in meiosis, it's a little bit different, okay? You have the parent cell, which has a full set of DNA. And then that DNA, it replicates here. Okay, so this is the replicated, this is the replicated version. This one's replicated. All right, and this one's the replicated version. And so the replicated version here is a little bit different. And so when it divides, it looks like this. I'll draw a better picture in a minute. And then when it divides again, you get 4 cells. And each of them you only see they have 1/2 of the chromosome here. So they have half of the amount of the DNA. There's 2 rounds of cell division, and each cell is genetically different. So I'm going to put a triangle on this one, a circle on this one, a square on this one, and a heart on this one, just to symbolize them as being different, okay? And we're going to go all through meiosis later, but I'm just kind of trying to broadly overview it here. So it's a modified type of cell division in sexually reproducing organisms, has 2 rounds of division, but only 1 round of DNA replication, okay? 1 round of DNA replication, that's important. And so you get cells with half of the number of chromosomes as the original cell. Okay? And we see that here. 2 rounds, they're different, half the DNA. 1 round, full DNA, and they're identical in mitosis and meiosis. So meiosis is how we make sperm and egg. It's responsible for making our sex cells. Mitosis, I may have said mitosis there, but I meant meiosis. Sorry if I spoke that wrong. Mitosis is responsible for making our body cells, our somatic cells. So we do mitosis when we cut ourselves, okay, and we have to regenerate cells there. Or for when we grow and we need to grow new cells, or when cells need to be replaced just because they're old and need to be repaired. That's when you do mitosis. You do meiosis to make sperm and egg. So ladies, you do that 1 time a month. Your body goes through meiosis 1 time a month, okay, before you ovulate and release an egg. Men, you make a lot more sperm than that, and so your body is constantly doing meiosis, okay? And so that's a little bit of a difference there. So when we look at the DNA that's contained in our cells, DNA is made up of genes. And so a gene is a unit of hereditary information consisting of a specific nucleotide sequence in DNA or RNA in some viruses. So let's think back to biochemistry. Remember, DNA or RNA are considered to be nucleic acids, okay? And so nucleic acids have 3 parts. What are the 3 parts of nucleic acid? You ought to be able to say this in your sleep, and if you can't, I'm really disappointed. It's a 5-carbon sugar, a phosphate group, and a nitrogen base, okay? Those nitrogen bases, as we have talked about briefly in Unit 1, are A, T, G, and C. And so when we have those nitrogen bases strung together in an order, a specific order, then that makes up a gene. It's a series of bases, of nucleotides in our DNA, and that makes up a gene, okay? And so we have genes that code for all of the traits that we have. Whether you have blue eyes or brown eyes, it's encoded in your genes, okay? Now, genes are found in a specific spot on the chromosome. Well, I should draw my chromosome, called a locus. And so this is a specific spot where that gene is found. Now, remember, your chromosome consists of coiled up DNA, okay? Your DNA is coiled in here to make that chromosome. So that locus also corresponds to, if we were able to magnify this right here, it also corresponds to, this is a terrible DNA, a specific segment of the DNA, okay, would be a gene as well. So a specific spot on a chromosome is technically the locus, but that correlates to a specific place on the DNA. Okay, hope that makes sense. So when we, going back to meiosis, I'm jumping around a little bit here, so I apologize, but it all comes together and makes sense, I promise. So when we pass these genes down, in meiosis, we make these gametes. These gametes are our sperm and our egg, okay? Remember, we called our body cells somatic cells, okay? Well, our sperm cells and our egg cells are called gametes. And I think we use that term in mitosis. I don't remember for sure, but I think we used that term when we studied mitosis. And so these gametes, these sperm and these egg, are responsible for passing our genes down between generations, okay? Because you have your parent DNA up here, and then they make that sperm, and they make that egg here in this 4th step, okay? And that is the DNA that gets passed down to you, all right, through the gametes. So let's talk just a little bit about sexual and asexual reproduction, because that's really important in mitosis and meiosis as well. So mitosis can sometimes be involved in asexual reproduction. So we use mitosis in our bodies to regenerate cells for growth, okay, or for repair or something like that, replacement. Some organisms do asexual reproduction, and they do a process very similar to mitosis to reproduce themselves. It produces a clone. It produces an offspring with one parent. A single individual is the sole parent and passes the copies of all of its genes to its offspring. And so the offspring are an exact copy of the parent, called a clone. And so asexual reproduction is a form of mitosis. So we can call it budding. Budding is an example of asexual reproduction. Some organisms form little buds on the surface. It kind of forms you remember in apoptosis, it kind of formed like a bleb in that picture. I don't know if you can picture it in your head. I can picture it in my head. And then these buds kind of break off, and they go form like their own new organism. And so that's an example of asexual reproduction, mitosis used to reproduce the whole organism, okay? Not just a cell, but the whole organism. But for us, we do sexual reproduction. And so we have two parents that give rise to an offspring with a unique combination. So they're not a clone. We have unique combinations from both parents. And so the offspring are genetically variable to their siblings and to both parents, okay? Now, somebody's going to ask me about a twins, because you're like, not all offsprings are the same. Maybe there's a twin. So what happens in a twin? So we know that twins can be fraternal or identical, okay? So we've got the egg and we've got the sperm, okay? Now, normally mom will release one egg each month, sperm will fertilize egg, and you get one baby, right? Now, in the case of fraternal twins, let's do that one first. Sometimes mom can release two eggs, and so you have two sperm that fertilize two eggs, and that would be an example of fraternal twins, okay? Sometimes mom will release one egg, but something happens that causes it to split, and then two different sperm can fertilize it, and that would be fraternal twins as well, okay? Now, identical twins is if you have one egg released, one sperm that fertilizes it, but then it later splits, and so they have identical DNA. Or if it split again and you had triplets, and you would have identical triplets, okay? And so that's how twins occur. But generally, for the most part, offspring of sexual reproduction have genetic variation both to their siblings and to both parents, okay? Also, every sperm and every egg is different from one another. And that's why we see that difference there, okay? So in one round of meiosis, as we saw over here, let me just go back to this diagram. This one had a circle, this one had a triangle, this one had a square, and this one had a heart, okay? So if this egg was fertilized, it would be a very different offspring than if the second egg was fertilized, okay? So every egg is very different, or every sperm is very different. So many times in genetics, we use these tools called karyotypes to study chromosomes and to be able to look for genetic disorders. And so what a karyotype is a picture of the chromosomes. And in a karyotype, they're always arranged by size. And so we start with number 1 is the biggest chromosome, and then the last one is the smallest set of chromosomes down here in number 22. Now, in humans, we have 23 pair or 46 chromosomes. So you see here we've got the 22 pair here, plus this one makes the 23rd pair. And so those first 22 pair are referred to as autosomes. They are non-sex chromosomes, okay? That last pair right here is the sex chromosome. So can you look at this and tell me if this is a boy or a girl? There's an XY. An XY is a boy. An XX is a girl. What you may notice about the chromosomes is the Y chromosome is much smaller than the X chromosome. The Y chromosome has much fewer genes on it than the X chromosome. Okay? So karyotypes can show us some genetic disorders, but not all genetic disorders. So for example, trisomy 21. Have you heard of it before? It's also known as Down syndrome. Trisomy 21 means it has or 21st chromosomes. And so somebody with Down syndrome, if you saw their karyotype, you would see another chromosome right there on number 21. Okay? Now, one thing I will say is that you don't take a picture of cells and get a perfect karyotype that looks like this. These have to be generated. And so basically what they do is they find cells that are in mitosis, and they freeze them and they manipulate the chromosomes to put them in a karyotype. So they don't look like this in the cell. But they are used to display genetic disorders. So you could see a chromosomal genetic disorder in a karyotype. Some genetic disorders also cause deletions in a chromosome. And so you may have a whole segment of a chromosome deleted. And so like for a number 7, for example, let's say you had a chromosome 7 deletion and this whole series of the chromosome were deleted. And so that chromosome would appear much shorter. Basically, this part would be chopped off and just added to this part. And so it would be much shorter. And so you would see that in a karyotype. But a karyotype is used to show genetic disorders. So what we see here in this karyotype is we have a replicated homologous chromosome. We're going to talk about what that word homologous means in just a minute. But this is a replicated chromosome. So let's go back, to the diagram that I'd kind of started a while ago of mitosis and meiosis. Okay, so in mitosis, when we started, we had a cell that had chromosomes. Okay, there's our one set of chromosomes. And it replicated, so then let's just draw it. It's kind of like an extra set there. Technically, it would look more like this, but I can't really draw it very well with this pencil and whatever. So, and then when it split, then it would look like this. And you have two set of identical chromosomes. In meiosis, you start with a cell that has this, okay? One of these came from mom, one of these came from dad, okay? So then they replicate, copy the one from mom, copy the one from dad. So when they divide, then you've got this. And then when they divide again, then you only have half. Okay? So you see we only have half of a chromosome in each one. It's not got the other side. And that's important because then you get the other half from the other sex chromosome. So if this is the sperm for the boy, then you're going to get the other half from mom when they come together in the egg in fertilization. Okay? And so that's how it has half of the DNA. Okay? So these are our sister chromatids. You see the sisters after they have been replicated. Okay? They're joined together at the region in the middle called the centromere. We talked about those in mitosis. And then this is a homologous chromosome. Okay? So we have the portion from dad and the portion from mom. Okay? homologous chromosomes, both sets of the pair. All right. Now, one other thing I wanted to say about this karyotype that I left off just a moment ago and came to me. I said that not all genetic disorders can be seen on a karyotype. So some genetic disorders are just a very, very minor change in the DNA, okay? For example, sickle cell anemia. Sickle cell anemia is a disease of the red blood cells. Normally, red blood cells are nice and round, but in sickle cell anemia, there is a change in one base, one letter, one A, T, G, or C in the DNA code that causes the shape to go from being perfectly round, where it is very lovely for carrying oxygen and moving seamlessly through the blood vessels, to being sickled, which looks like about the shape of a banana, okay? Which doesn't carry oxygen well, also doesn't move through the blood vessels very well. But that's a change in one base. So let's say, for example, I don't know the sequence, but I'm going to call it this. Let's say this makes a normal red blood cell. A sickled red blood cell is just one change. It's one letter that got screwed up, okay? So that is a genetic disorder. It can be inherited or passed down, but that is not something that you would be able to see in a chromosome. You can't see a single gene mutation, okay? That's a single gene mutation. This is the gene that controls the shape of the red blood cell, which is in one spot on the chromosome, one locus on the chromosome. And so you can't see that on a karyotype. A karyotype shows chromosomal mutations, big mutations at the chromosomal level, okay? Last slide here. So this shows our homologous chromosomes. And so this is a homologous pair. When you've got the set from mom that's been replicated and the set from dad that's been replicated. The one from the mom plus the one from the dad makes the homologous chromosome, or sometimes they're referred as homologs, okay? And these are replicated. So that's a homolog. These two, if they were bound up together, they would be referred to as homologs. And then these two are homologs, okay? These are sisters. These are sisters. These are sisters. And then these, showing here in this diagram, are non-sister chromatids. So non-sisters are going to be the, in a homologous pair, the ones that are not paired together. Those will be referred to as non-sister chromatids. Okay? So, looking at this cell, we see what's referred to as haploid numbers and diploid numbers. And we'll talk about that more in just when we talk about meiosis later this week. But N equals the haploid number. 2N equals the diploid number. The haploid number is how many are in the sex cells, okay? Our haploid number is 23. We have 23 chromosomes in our sex cells, in our sperm and in our eggs, okay? Our diploid number is 46. That's how many are in our body cells, okay? The full number of chromosomes. The haploid number is half the number of chromosomes. And so here, N equals 3, okay? That means in a sex chromosome, they would have 3 chromosomes. And then that means that the diploid number would be what? Should say the diploid number would be 6 for this organism, okay? And different organisms have different numbers of chromosomes. Now, some organisms will have the same just because there's only a certain number of chromosomes. We are probably not the only organism with 46 chromosomes, with a diploid number of 46. But chromosome number does vary from organism to organism. And it's also interesting to note that more sophisticated organisms, for example, don't necessarily have the most chromosomes. There are plenty of organisms that have many more chromosomes than humans. So size doesn't dictate it. Level of sophistication of the organism or anything like that doesn't dictate it, but it is variable from organism to organism. Okay, so that concludes the lesson today on genetics. If you have any questions, let me know, and if not, then you all have a great day.